Hello and welcome. The goal of this talk is to provide advice for conservationists wishing to make a seed collection. My name is Sean Hoban, and I'm a conservation biologist with more than 10 years experience in the genetic aspects of plant conservation. I work at the Morton Arboretum, a botanic garden and research institute near Chicago. I will start with an introduction into the uses of seed collections and why they should be genetically diverse. I will then talk about how to determine how many and which populations to sample and how many seeds to sample. I will conclude with considerations while in the field, including data collection and timing of seed collection. I will talk about seed collections in a generic manner, so that this presentation will be useful for rare and common species. Your collection might be for a seed bank, or living plants for a botanic garden, or germplasm collection, or other purpose, but the same rules apply for seed collection. A small note, some plants might be easier to collect vegetatively, so even though I might say seed collection, you might be collecting other plant parts. First, let's talk about seed collections and genetic diversity. A collection of plants kept in a seed bank or botanic garden might have different uses, and might have multiple uses over time. First, we might think of long-term conservation of a rare species in case it goes extinct in the wild, a safeguard against extinction. Another use of collections is for scientific research, including research into seed germination ecology, pollination biology, evolutionary biology, dispersal, etc. Interestingly, sometimes this research will provide information directly leading to better seed collection and care. A third use of collections is to provide seed or whole plants for species recovery actions, such as establishing new populations in the wild, helping increase population size, or restoring habitats. Genetic diversity is a principal concern for any of these uses of seed collections. You want to make every seed count. You want to get the most amount of genetic diversity in the relatively small amount of seed that you can preserve. Why is genetic diversity important? Genetic diversity helps species avoid inbreeding and provides the breadth of diversity needed for species to adapt to new environmental conditions. Especially important as species face climate change, land use change, invasive pests, and pollution. Genetic diversity in common species also helps promote stability and species diversity of the overall ecosystem. Lastly, genetic diversity is often useful for humans for improving agriculture, forestry, and horticulture by providing new forms, resistance to disease, or ability to grow in new conditions. So, what do you as collectors need as prerequisites before you make a seed collection? You want to make sure you can care for that seed for the long term. I emphasize that if you get seed and don't care for it, it can be wasted effort. I know from first experience how frustrating it can be to watch many new seedlings die. You need space, people with time to care for seeds, technical facilities such as cleaning equipment, freezers, etc., money for supplies, and a long-term plan. You need botanical expertise to identify the species in the field and know if you have the seed at its highest level of maturity so that it will be viable. For example, shown here are immature and mature acorns. You need knowledge of how to care for that species, such as its preferred soil type, storage and chilling requirements, and germination conditions. If you don't have these prerequisites, resources can be spent to prepare for future collections. You can spend your time and money working on improving equipment and infrastructure, forming collaborations with other gardens with whom you can share the effort and share expertise, or performing the basic research necessary to gain knowledge that will help your species of concern, such as collecting some seed to test germination requirements. All this work leads to seed conservation in the long term even if you are not able to do seed collections immediately. Shown here are logos of several organizations who can help provide partnership or other support. Now, let's talk about choosing populations. Sometimes a species occurs in only one population, but more often there are multiple or many populations, sometimes across a large geographic area. Shown here are seed collection sites from a recent collection I made of Shinnery oak, or Quercus havardii, which occurs across at least 5 different states in the United States. The first question is how many populations? You want to sample from enough populations to allow for adaptation and persistence under current and future climates. For example, Shinnery oak occurs in diverse environments like those shown here, and you want to make sure that this environmental diversity is represented in your collections. Another example of a very extensive seed collection was reported in the Public Gardens magazine in 2017. The Brenton Arboretum has made more than 80 seed collections across the range of the Kentucky coffee tree, shown here in orange, an uncommon species in eastern North America. These collections even cover most of the outlying or disjunct populations of the species. This table provides a very high-level summary of how many populations you might aim for, though you should also consider your goals and the purpose of the seed. For rare species, all populations should be visited, especially if fewer than 5 populations are in existence. For any species, if the species occurs in different habitat types, hardiness zones, or other ecological zones, at minimum, 1 or 2 populations from each of these ecological zones should be sampled. The more populations there are, the more should be visited. For example, a common species might be sampled at 20 to 30 locations to get most of the genetic variation. Partnerships may be needed for such large-scale collections. Again, keep in mind the goal to ensure capture of environmental adaptations and get enough genetic variation to minimize inbreeding. Now, how to choose which populations to sample from. The basic rule is to sample with wide spatial coverage. Sampling evenly across a species range is an excellent start, as in the example of Shinnery oak or the Kentucky coffee tree that I just mentioned. There are 3 more sophisticated ways to choose populations, and I will explain each. Using ecological maps, using a genetic data survey, and using genetic models. First, if you have maps of ecological regions or habitat types for your country, such as the USDA hardiness zones or ecoregions defined by the EPA or Forest Service, you can overlay these maps on your species range map or occurrence points. Then, using this overlay, you choose populations to sample such that you cover each of the ecoregions with your sampling. For example, shown here are occurrence points in white triangles of Quercus lacei, another oak species, in Texas, United States, overlaid on the level 4 ecoregions, that is, the different colors. You can see that the species occurs in 4 to 5 ecoregions. Existing samples, the black triangles, however, only cover one ecoregion. Sampling 1 to 2 sites at minimum from each of the unrepresented ecoregions is recommended. If you have resources to do so, you can perform a genetic survey in which you assess the genetic diversity in each population, and then choose the most diverse or divergent populations to sample. This will cost around $5,000 to $10,000 US and take 6 months to a year of part-time work, depending on the species. A genetic survey consists of taking leaves or other tissue from as many individual plants as possible from all known populations. Typically, this is done for rare species where most individuals can be sampled. You then genotype or obtain DNA sequences from these samples. Analyzing the DNA will then reveal the most genetically variable or distinct populations to sample from. It can also inform the collector about what seed could be mixed together in some future use of that seed. An example of this work is that of Patrick Griffith, who has analyzed several palms and cycad species, including Zamia decumbens, which is shown here. This graphic shows the genetic variation of two wild populations and the ex situ populations, which are shown to well represent those wild populations. If genetic data cannot be obtained, a genetic model can be used for some species. This approach is used to predict where the most genetic variation is likely to be and to test different sampling strategies. It is not as accurate as having actual genetic data, but it can be cheaper and faster. I have recently done this for European ash, which is threatened by a devastating disease. I used surveys of ash population abundance along with knowledge of ash tree biology to build a demographic genetic model that predicts where genetic diversity is most likely to be high in populations across the UK. I then tested how different seed sampling strategies, including number and location of target populations and number of seed, using this model. I also evaluated the current seed collection to see how complete it is. We determined that current seed collections already capture more than 90% of the alleles of this species. This work is published recently in Biological Conservation, and there are many more findings and recommendations in this paper. A few additional notes on populations. If you have a choice, it is best to sample from populations with large size and a history of stable large size. These populations are most likely to be locally adapted and to not show inbreeding. Once a population has started to decline, it will often become less adapted. An interesting proposal that I have seen is to also consider taking some of your samples in non-pristine sites, such as fallow agricultural land, on poor soil, in abandoned lots, etc., as these may have unique diversity and useful adaptations for the future. For more information on what choice of seed can lead to successful restoration outcomes, see this citation from 2011. Now we visit the question of how many plants to sample from and how many seeds to collect. Some plants, like Fraxinus shown here, produce many thousands of seeds. The best minimum sampling is to sample from at least 50 randomly chosen plants throughout the population and to take several, usually at least 10, seeds per plant, which thus means at least 500 seeds per population. If the population is smaller than 50 individuals, it is suggested to sample from every plant. However, sometimes this is not enough, because some plant species show high rates of seed infestation or empty seed, low germination rates, or high seedling mortality. Seeds are also used for testing and research. You should adjust your seed collection plan to compensate for expected losses. For example, if 50% of your seed fail to germinate, collecting 100 seed means you really only have 50 usable seed. You would need to collect 200 seed to have 100 usable seed. That is how you compensate for expected losses to increase the amount of seed you collect. If you have such data, such as from research that has been performed in your target plant species or genus, you should use this data to plan your collection. This citation, Cochrane et al. 2007, provides some useful examples for real species. Some authors suggest assuming 90% losses from seed to adult plant, thus collecting 10 times as much seed as you think you will need. I emphasize here that seed banks and restoration programs should keep data on how seed is lost over time, because that information will eventually be useful for the next round of seed collecting. Lastly, I want to emphasize that these are best practices, but if a seed collection must be smaller than ideal due to logistical or biological constraints, it does not mean it is not an important seed collection. Sometimes, even small collections have good value. The next point is that it's not just about the number of seeds. How you sample is also an important consideration. Plants must be chosen as randomly as possible or along an evenly spaced grid if you want to maximize diversity and minimize inbreeding. The minimum sampling that we have been discussing so far assumes random sampling. This picture contrasts random and grid sampling with some other types of sampling. Most important is to avoid sampling plants that are all adjacent to each other, as in the upper left. These plants are all likely to be related. In forest trees, a common guideline is to allow at least 100 meters between trees sampled for seed. You should also take the same amount of seed per plant. That is, do not sample all or most of your seed from one plant and only a little from other plants. Make it even. If this advice is followed, the seed is more likely to be diverse and a good representation of the source population. I will now discuss some important practical advice for after you have chosen your populations and determined how much you need to sample. BGCI has other presentations about other aspects of seed collecting that I will not cover, including equipment needed, how to plan logistics and get permits, and cleaning seed. So please visit this link for more valuable guidance. It is important to leave enough seed in the population for natural regeneration and for forage for wildlife that might depend on that seed. A typical safe minimum is to take no more than 20% of the seed available in a population at a given time. If you know much about the species life cycle, you can use demographic modeling to determine a safe harvest percentage. It might range from being able only to take a few seeds to being able to take most of the seed. But again, on average, 20% is a good maximum amount of seed you can take. You can see Justin Meesen's publication from 2017 about how to use stage-based demographic models to determine safe harvest levels. In some cases, however, if the species is in imminent danger of being lost in the very near term, such as on an eroding cliff, you may wish to take all available seed. Of course, timing the collection so that the seed is at its most ripe is very important to ensure high germination rates. Seed that is underripe may not germinate at all. However, in many plant species, not all seed will be available and ripe at the same time. This is important because it is known that seed ripening and phenology, flowering, has some genetic basis. It might be heritable. If you only sample at one time point, you lose some of the genetic diversity underlying this key trait, and your restored population would have a shortened period of flowering and fruiting, which will ultimately impede the species' ability to survive events like early frosts or droughts. Sampling throughout the season captures more diversity and increases the species' chance of persistence. So you should sample repeatedly throughout the fruiting season, again focusing on mature seed. You should also sample across multiple years because in some years, some individuals won't produce any fruit. Last point, but not least important, is data. Seed without data on species ID, place of origin, and other important information is almost useless. At minimum, the data sheet should record species name, GPS location, the number of plants sampled from, the number of plants existing in the population, and the sampling approach. By this, I mean, did you use random sampling? How much distance was between plants, etc. If possible, also record data on plant size and health, environmental characteristics of the site, historical uses and threats at the site, and the nearby plant community. All this will help in the eventual use of the seed, where and how it can be used, such as where it should be introduced. Also, please collect data on germination rates and loss of seed over time. As mentioned earlier, this will help guide how much is needed for future seed collections. You don't have to make your own field data sheets. You can use one of several that are available online. Just search for these very informative documents. I will now try to summarize all of this information in the next two slides. First, you determine your seed collection goal, what will be done with the seed. This choice affects how much you need to collect. Then, you determine if you have the resources, equipment, long-term storage, and knowledge. If not, you can share the work with other gardens and still help seed conservation. Next, sample at least 5 populations and in different ecoregions across the species range. For common species, this might be increased to 20 to 30 or more populations. Within each population, sample at least 50 plants with 10 to 100 seed per plant. This number should be adjusted, however, based upon what you know about seed losses, such as germination rates, etc. For many species, it may need 500 to 5,000 seed or more per population. Next, make sure you are sampling as randomly as possible when you select plants to sample seed from. Next, make sure that you sample throughout the fruiting season and over multiple seasons. Also, please do not overharvest. Take 20% of the seed available as a basic maximum. However, this could be adjusted based on what you know about the species biology. Lastly, keep as much data as possible in the field and afterwards during seed use, and if possible, share this with others. Thank you for listening to this talk. I hope the examples, advice, and references have been useful. Good luck in your seed collections.